How do pollen and phytoliths reveal ancient environments? Imagine tiny clues hidden in the soil that can tell us what the world looked like thousands of years ago. That's what pollen and phytoliths do for archaeologists. They are microscopic plant remains that help us piece together past landscapes and climate conditions. Pollen grains are the male reproductive cells of flowering plants. They are produced in huge amounts and have unique shapes depending on the plant species. Because of their durability, pollen can be preserved in soil layers for thousands of years. When archaeologists find and identify different pollen types in a soil sample, they can figure out what kinds of plants grew nearby. This helps us understand the environment at different times in history. For example, if a soil sample contains lots of pollen from trees like oak or pine, it suggests a forested landscape. If grass pollen dominates, it might point to open plains or grasslands. The age of the soil layer can also be estimated based on how well the pollen is preserved. Phytoliths are tiny silica structures that form inside plant cells when plants absorb minerals like silica from the soil. After the plant dies, these silica bodies stay behind because they are very tough. Different plants produce phytoliths with specific shapes and sizes, so scientists can match these tiny silica structures to particular plant types. Because phytoliths are so durable, they can survive in soil for thousands of years, giving us a snapshot of local vegetation. If archaeologists find phytoliths from domesticated grains like rice or millet, it can indicate early farming activities and the types of plants people cultivated. Combining pollen and phytolith analysis gives a fuller picture of ancient environments. Pollen can show regional vegetation patterns, while phytoliths often reveal what was growing right around the site. These remains can also tell us about environmental changes caused by natural shifts or human activities like clearing land or farming. To analyze these tiny clues, archaeologists collect soil samples from specific layers in a site stratigraphy. They then extract the pollen and phytoliths and examine them under microscopes. By identifying the plant species and understanding their context, researchers can reconstruct how the environment changed over time. This process helps us see how ancient societies adapted to their surroundings and how landscapes evolved. So, in essence, pollen and phytoliths act as microscopic time capsules, revealing the story of past environments through the plants that once thrived there.